Hi, everyone. Welcome to the final Take Action webinar in our series, Week 7. Thanks for joining us today. This series is designed to bring you valuable information on weed and herbicide resistance management topics. It's a collaborative effort with the Take Action program and several land-grant university weed scientists. Joining us week, we have some great speakers. We have Dr. Mandy Bish from the University of Missouri. Hi, Mandy. How are you?
I'm good, Mark. Thanks.
Good. And then we have from Purdue, actually, Bryan Young speaking. Bryan, how are you?
I'm doing well. Thank you, Mark.
And then also on the call, we have Bill Johnson, who's one of the co-organizers with me on this. I'm Mark Locks from Ohio State, if I didn't already say that. A little more information on the Take Action program. Take Action, farmers' freedom to operate is being threatened by the increase in spread of pesticide resistance. The consequences include short and long-term economic challenges, decreasing land values, the uncertain regulatory pathway to access crop protection tools, crop losses, and other challenges. Take Action is a farmer-focused education platform designed to help farmers and their advisors manage herbicide, fungicide, and insect resistance. The goal is to encourage the adoption of management practices that lessen the impact of resistant pests and preserve current and future crop protection technology. Take Action is brought to you by the Soy Checkoff. For more information on Take Action, you can always visit the website, which is www.iwilltakeaction.com. As a reminder, you can submit questions through the presentation. As I said, as we were leading up, our question box on the platform isn't working very well here. So you are welcome to submit questions at any time via Twitter. If you're getting information on this via that, then you'll know how to do that. And you can also email questions directly to the moderator, co-moderator today, Bill Johnson, WGJ at Purdue.edu. Our first presenter is Dr. Mandy Bish. Mandy is an extension specialist at the University of Missouri, where she has worked with state specialist Kevin Bradley since 2014. She has studied temperature inversions since 2015, when the question, how might inversions affect ground pesticide applications, arose. With guidance from climatologists at Missouri and North Dakota State University, the group began monitoring inversions at 3 locations in Missouri. Since that time, and with funding from USB, Missouri Soybean Merchandising Council, and Kentucky Soybean Board, the Inversion Monitoring Network has expanded to 29 stations. Mandy is responsible for analyzing the generated data for inversions and other weather factors that might associate with off-target pesticide movement. Mandy grew up in a farm in central Missouri before earning her Bachelor of Science from the University of Missouri and then her Ph.D. at the University of Maryland. Welcome, Mandy, and I'm going to turn it over to you. You should have control.
Great. Thanks, Mark. Good morning, everyone, and thanks for joining in. As Mark said, my name is Mandy Bish, and I'm going to spend time talking about hopefully 3 things today. The first being inversions. The second will go into a little more detail on weather and off-target movement, not related to inversions. And then third, if we have time, I'll talk a little bit about some USB work that we're doing, or sponsored work that we're doing on Rainfall and Dicamba. Okay, so what's the evidence that inversions contribute to dicamba off-target movement? Well, one is field observations. The timing of some applications, which were in the late afternoon, thinking that you're doing good, waiting for the wind to die down, and patterns of injury in the field. So when you see consistent injury in the field, when you see injury in low-lying spots in the field, consistently throughout a field, those are both possible indicators that an inversion could have moved the dicamba. Another source of, I apologize, having trouble moving the screen. So another thing that we did to look at how inversions might move dicamba is we actually sprayed during inversions. And so that's what this graph is showing. We sprayed during inversion conditions, and then we sprayed during the middle of the day. And this is a summary of 10 experiments. And the black bar is when we sprayed during inversions, and that shows how much dicamba we collected in the air the 8 hours after we applied the dicamba. And the gold bar is showing the midday application. So we found that when you spray during an inversion, we got about 3 times the amount of dicamba collected in the air than compared to when we sprayed midday. So those are both kind of evidence of inversions contributing to dicamba off-target movement. So what are inversions? And basically, it's just when the air temperature is inverted. On the left, you can see conditions when there's no inversion. So warmer air is near the Earth's surface, and this is due to sunlight hitting the Earth's surface and warming that air. Well, we all know from the winter that in our houses that warm air rises and cool air sinks. And so as warm air rises, the cool air sinks and it gets warmed, and you have this constant shuffling of air when there's no inversion present. And usually there's cumulus clouds at the top of this shuffling column, and you get wind as the warm air and cool air mix. Well, when an inversion occurs, you get inverted temperatures, that flips. And so the cool air is on the bottom and the warm air is on the top. And what I've shown here is a radiation inversion. So when the sun sets, there's no longer warming of that air near the Earth's surface. When you have cool air on the bottom and warm air on the top, the warm air is not going to rise, and so that air mass is very stable. Now, there are other conditions that can cause inversions or that cool air on bottom, and those would be like cool air drainage. You know, cool air is more dense, it drains. If you have valleys, then air moving across the tops of the hills or the mountains can cause friction, which is going to cause the air on top to be warmer. So there are multiple causes, but inverted temperatures are anytime there's cool air on the bottom and warmer air on the top. And what this does is it kind of creates a cap, so there's no mixing between that cool air and warm air boundary. And I illustrate this with a smoke bomb because it's a good visual. So we released a red smoke bomb in the middle of the afternoon when there was no inversion present. And if you look closely, you can see the red at release. And then 50 seconds later, you don't see any of the smoke bomb left. It's all dispersed. There's no inversion present. But then when we went back the same evening and released a smoke bomb, again, you can see the red cloud at release. 50 seconds later, you can see that that smoke bomb has not dispersed. So the stable air mass is keeping it from dispersing. There's no wind to disperse it. And what's going to happen is that smoke bomb is going to go to the red is going to go to the top of that line where the cool air meets the warm air. And some of you have probably seen this, like if you've driven by a smokestack or a fire and seen the horizontal movement of a cloud or of a cloud, then that would be the top of that boundary line. So how common are inversions, and are they something that we really need to worry about? As Mark mentioned in the intro, we had started with 3 stations in Missouri, and then that has expanded to 29 stations across many areas of the soybean growing region. And one of the things I want to point out about our inversion monitoring is if you look at the right, we're monitoring air temperatures at 3 heights, and those 3 heights are relevant to ground pesticide application. So there's been a lot of work with this, and aerial pesticide applicators have known about inversions and their problematicness for a long time, but there hasn't been any work done to our knowledge prior to this on ground pesticide applications. So as you can see, 10 foot is our highest, and then we go down to 1.5 feet for monitoring inversions. And so in 2019, this is just some of the 29 sites. These were the number of evenings in which we had inversions. So the black bars are number of evenings in June, and the gold bars are number of evenings in July. Now there's a couple of things I want to point out about this. One is that 2019 was pretty wet in May and June. Wet conditions indicate that there was cloud cover, so there weren't as many nights maybe as we would usually anticipate that had inversions. Cloud cover is going to interfere with inversion formation. The second thing is that these are conservative estimates. So what I mean by that is we take into account the accuracy of the temperature probes and some other technical aspects. So anything that I'm calling an inversion on this graph, I'm confident is an inversion. There's likely more inversions than what I'm showing here. And part of the reason for that is when we go on to doing some of the forecasting, we want to make sure that what we're calling an inversion is an inversion. So I would almost say this is the minimum number of inversions that or minimum number of days where inversions formed in 2019 for these locations. And we saw inversions at all locations, I should say that as well. And so this is kind of what I was talking about with you have to pay attention to what the conditions were one year versus another year. This is just an example of a 3-year trend of inversions taken from the southeastern portion of Missouri. 2015 in Missouri was really was a wet year in the spring and early summer. And you can see those dark gray bars. Those were the number of days each month with inversions, and it was less than when you compare it to 2016 and 2017. 2017, we had an issue with our pollinator. We had a pollinator plot that was shading our sensor, and so we didn't look at that data, which is why there's no June 2017 data there. But you can see that it's important not just to take 1 year and make assumptions on it, that you need multiple years to really understand. Another thing I want to point out by this is we have submitted this work to climatologists. Obviously, I'm a weed scientist. My colleagues are all weed scientists, but we've worked with climatologists and they accepted this work on inversions. So we feel pretty confident. I feel confident that what I'm calling an inversion is an inversion. And as we talk about indicators of an inversion, so how do we watch out for an inversion now that we know that they're common? The new Dicamba labels say that you must cut off applications 2 hours prior to sunset. Well, what we're learning is that sunset may not be the best predictor at all geographies. So what I have here is the average time that an inversion began forming. These are nighttime hours, so 7:40 p.m., 8:20 p.m. in Indiana. Why is Tennessee highlighted? Because at the Tennessee sites, first of all, you can see the difference in the timing, and I'm going to talk more about this later. But those sites had inversions that began forming prior to 2 hours before sunset. So the inversion would begin forming before the label restriction times. And if we look closer, this may be more of a mid-south problem, potentially. Again, some sites we only have 1 year of data, so I don't want to say that too strongly, but we have a lot of sites where inversions are beginning to set up prior to that 2 hour cutoff window. And again, I want to point out this Tennessee site. As you can see, there's an hour difference between the bottom and the hill when inversions form. This has over an hour difference. And then in August, it gets closer, but this is after 2 years of data for most months, not May. And what we know is that these two weather stations are actually less than 1 mile apart from each other. So you have two weather stations that are less than a mile apart from each other, and you have inversions forming an hour different, an hour earlier at one site than the other. There's less than 100 feet elevation. And so what I like to use this example for is that inversions can set in different fields at different times. So topography really is going to play a role in when you could be spraying one field, then you could drive a mile down the road and there could be a field where an inversion has set in. In the Tennessee bottom site where the inversion set in earlier, there's a tree line that sits to the southeast, and prevailing winds during the summer come from the south. So one possibility is that tree line is blocking wind. If you don't have wind, then inversions are going to form a little more quickly, likely. And also it's lower, so the cool air is more dense. It's going to settle in the lowest areas. So knowing your surrounding is important when it comes to inversions. And I like to tell our applicators that right now, forecasting apps aren't quite there. They may get there for some geographies and they may not get there for other geographies. There are some fields that are just going to be really hard to predict. So it's really important to know the indicators of an inversion. Clear sky, so when the clouds begin dissipating, and lack of wind are two of the best indicators. Dew or frost and low-lying fog are both indicators, but I don't like those as much because if you sprayed the night before and then you see dew or frost or low-lying fog the next day, then it's a little too late. So, you know, as soon as those winds begin dying down, when you think it would be an ideal time to spray, that's probably a sign that an inversion is forming and it's not an ideal time to spray. I'm just going to take a couple minutes longer and talk about something else that with the weather data that we've been generating. And so this is that graph I showed at the very beginning. Again, the black bars are dicamba in the air that we, after we sprayed during inversion conditions. So we made an off-label application and we knew it to see how much dicamba was in the air. Then we came back and sprayed during the daytime in a labeled condition. And what we find is that for the days after that application, so this is 24 to 48 hours after application and 48 to 72 hours after application, that we're still detecting dicamba in the air at similar levels. And so what's causing that? And this is just showing another way that we're using this data. Basically, these are 8 of our sites, of our 29, and average air temperatures through May, through June, July, and August. And what you can see is that basically the punchline is we do see temperatures above 85 degrees Fahrenheit commonly in most of our locations. And we know that 85 degrees is kind of the threshold for volatility. But one thing about air temperatures is that, you know, in some areas that do not have the same issues of volatility, the air temperatures are very similar. So that leads me to think that it's not just air temperature. The air temperature leads to volatility, but there has to be something more. And wind speed is another thing that we're looking at. And this is, again, just 8 of our locations. And what we see in these regions is that wind speed is, we don't get that high of sustained winds. So 10 miles per hour at each of these. We're not seeing wind speeds that sustain higher than 10 miles per hour. And so what we're working on now to try to understand is, do we have enough wind to disperse all the dicamba being applied? And I think I'll just leave with this point. On the left is an EPA generated map of annual rate of herbicides. So the redder the area, the more herbicide being applied in pounds per acre. Okay, and you can see Illinois, Indiana, this region right here, a lot of red, where we have some of our biggest issues. On the right is a chart, again, EPA generated with wind speed. So initially EPA was using this chart to say, hey, these are places where it's too windy to spray. But what we took it for is, hey, where are places where we don't have a lot of wind? And those places are in blue. So a lack of windiness, so to say, is in the blue. So if you overlay the blue areas with the red, you can see that maybe we do not have enough wind to disperse all the dicamba, and that's what we're working on right now. For the sake of time, I'm not going to go over this slide in detail, but USB has sponsored some work for my colleagues and I, and to look at rainfall following dicamba application, and our preliminary data does suggest that rainfall can help reduce volatility. And so with that, you know, inversions are common across regions during the growing season, and we know that dicamba will be suspended in an inversion if it's sprayed during one. Inversion conditions can vary between fields, so it's important to know the indicators of an inversion. Cool air moves to the lowest point, and it's going to transport the dicamba in it. So again, if you see a lot of injury in a field that's in low-lying areas, we typically associate that with runoff, but it might be that it's been moved by inversion. And we may need wind to help disperse dicamba that volatilizes. And then rainfall after an application seems to reduce the amount of dicamba. So with that, I'm done, and I can hand it back over to Mark.
Great, thank you. We're going to hold questions till the end. I know I thought of a couple right off the top of my head, so good presentation, thanks. I will try to get this advanced. There we go. Our second presenter is Dr. Bryan Young. Bryan is a professor of wheat science in the Department of Botany and Plant Pathology at Purdue University. Bryan earned his bachelor's degree in crop and soil science at Michigan State University and doctorate in crop science with a specialization in wheat science from the University of Illinois. Bryan has held an academic appointment with teaching, research, and outreach activities for over 20 years. His research and education focuses on herbicide application technology and characterizing herbicide resistance in weeds. I will hand it off to you, Bryan. Thanks.
All right. Well, thank you, Mark, for the introduction, and good morning, everyone. Mandy did an excellent job presenting the whole issue of off-target movement in force and the temperature inversions. And really, when we talk about temperature inversions, it's these inversions that can help move some of the volatile herbicide, in this case dicamba, to places that we don't want to go, the sensitive areas. So, it's a good lead into what I'm presenting today. So in terms of overall off-target movement, we have different causes that we're all familiar with. The physical spray drift, which, really the label addresses fairly well with the buffers that we observe, the environmental conditions at the time of application, the nozzles and drift reduction agents that are involved. Sometimes it can occur through tank contamination, just not cleaning out the tank fairly well, or mixing errors can occur as well. And then volatility. So these are not necessarily placed in terms of importance or order, but I guess I would suggest that volatility is probably the least of concerns regarding all three of these factors. But volatility is something real that we have observed, and obviously we've done research along those lines. So what influences volatility? Well, obviously it has to do with the chemistry in the spray tank, chemistry of the dicamba formulation itself, and some of the different salts available, but also the environmental conditions might lend itself towards more off-target movement of that vapor as well, kind of like Mandy suggested in some of the temperature inversions. So we designed some research with United Soybean Board to look at some of the chemistry in the tank. This came from some growers on the USB board who had some questions about, what are things that we can do in terms of things in that spray solution that might be influencing volatility, at least to any extent. So we started with 3 formulations of dicamba. Clarity, which is not labeled, of course, for extended crops, but it's kind of the standard that we've been using in corn for quite some time. And then the improved formulations for use in extend crops, Xtendimax and Engenia, and we applied all those at a 1/2 pound acid equivalent, which is the standard use rate in extend crops. We had the soybeans, as you can see to the right, that we put into these tubs, but before doing that, we sprayed them in our research chamber with this dose of dicamba, 1/2 pound of herbicide active ingredient on V2 soybeans, using standard nozzles and 20 gallons per acre. So after they're treated, they were placed in these bins, and you can see that we have foil over top of the soil, the potting media of these pots. And the reason for that is, one, we didn't want any water to evaporate off the surface because the soil was wetted, of course, to keep the soybeans growing, but also wanted to make sure that we weren't measuring any volatility of dicamba off the soil surface itself. We wanted to measure it off the leaf surface. So that's the other reason why after application, we applied the tin foil, aluminum foil, to the tops of these soybean pots. So we then took these tubs that would have had the soybean plants in them and put them in a walk-in chamber, a growth environment that was, we had a 48-hour incubation time at a temperature, rather high temperature, 35 degrees Celsius, and 40% relative humidity, that those conditions are known to increase volatility. So high temperatures, and as Mandy said, we know it's not the only factor, but it's an important factor. And low humidity is when we typically see higher levels of volatility. So we set the growth chamber to environmental conditions that would favor volatility. And then we had air moving across these plants inside the chamber from left to right, as you see it oriented on the slide. And that was intended to move the air to an air sampler that was positioned on the right-hand side of the chamber. And so that filter essentially was collecting any dicamba that was in that air. And we wanted to make sure we had enough air exchange so that we'd have enough dicamba in the chamber air that we'd have on the sample puff plug, as we would call it, so that we could analyze later. So one of the first things that was discussed is, what about dirty water? Sometimes we have good water sources that we spray with. In other cases, it might be a little bit murky water. And so grower had the question about, what about when you have soil or other sediment that might be present in the spray water that you have? Now we'll just start out with this. We know from a herbicide efficacy standpoint that we don't get good weed control whenever we have soil sediment involved, because it can tie up your herbicides and not allow them to be absorbed and have their activity inside the plant. But are there other things that might be involved that might influence volatility of the herbicide? So we looked at different levels of clay and organic matter content. We developed these turbid levels, if you will, based off of, frankly, the Wabash River flowing through just on the east side of our campus spring of last year, which is pretty murky. And so that was worst case scenario that we wanted to test. And then we had high and low concentrations of both these to see if any level of turbidity would cause some problems with volatility. So the data I'm going to show you today is all going to follow a similar format. I have the 3 different dicamba formulations at the top. And I just want to mention this right now. The way that we designed this research, because of space limitations, we're not able to make comparisons of these 3 formulations across those 3 products. We're only making comparisons within each dicamba formulation, moving down in the column for the treatments. In this case, it'd be the turbidity levels, okay? And I'll remind you of that a little bit later, but we're not making comparisons, direct comparisons across these dicamba formulations. Just want to know, were these formulations influenced by our treatments, in this case, turbidity? And you can see that in this first case, clarity, clarity, we had a dicamba concentration in the air that we sampled of about 4,200 nanograms per meter cubed. From a statistical standpoint, none of the turbidity levels increased volatility or the amount of dicamba in that air. Now we have some numerical differences, but because of the sensitivity of our test system, we can't really call these significantly different from a statistical standpoint. They're just numerically different. And that's true for clarity, it's true for Xtendimax, and was also true for Engenia, that we didn't have any increases in volatility. Now you might notice that for Engenia, we did have a case where there was a slight reduction, at least from one high organic matter sample to a high clay content soil or turbidity. But again, none of these caused an increase in volatility compared to the herbicide in clean water by itself. So now that we answered that one, the next question was, what about pH? And there's research out there that would suggest that different herbicide solutions with dicamba can reduce your herbicide spray solution pH. And under lower pH environments, you can form that acid of dicamba, which is the volatile form of dicamba that we're concerned about. So in looking at the pHs of typical spray solutions, it might run from 3 to 8 for pH units. 8 if you have some fairly alkaline water supplies that you're working with. Now down to 3, you're not going to find that from any groundwater or surface waters, but we might get down to pH 3 when we add certain chemistry, such as adjuvants that are water conditioners or acidifiers for herbicide efficacy reasons. But we know, according to some of the dicamba labels right now, the way they're written is that if you have a pH of spray solution that's under 5, that's reason to be concerned that there might be increase in volatility, and you might want to adjust that pH higher. So I'm going to present these by each herbicide formulation, the first one being clarity formulation. And when we just added that to deionized water, the pH of the spray solution was 6.3. So what we would feel to be in a fairly acceptable pH range. But then we adjusted the pH using simple chemistry. So these aren't buffered pH solutions, but we just adjusted the pH down to pH 3, 4, and all the way up to 8. And you can see the dotted line, dashed line, is the amount of volatility, amount of dicamba we detected for clarity with no pH adjustment. So that's from pH 6.3. And what you can note here is that we had a difference between pH 3 having higher dicamba volatility and more was found in that air sample than at a pH 7. So there was a pH difference, but really pH 4 and 5 didn't cause any major impact compared to a no pH adjustment with that clarity formulation. Now, what we found is we're a little bit surprised by some of these results. So we want to know why in the world, pH 3 wouldn't have a more dramatic impact. And so after these results, we went back and repeated the research and looked at the surface pH of the leaf, our target. And what we found is even though we adjusted the spray solution pH to 3, the pH on the deposit of that spray solution on the leaf surface within a minute after application was 6. And when we started with pH 4 for spray solution, the pH of the deposit on the leaf was 6.5. And so you can see that the pH of these deposits on leaf surfaces, soybean leaf surfaces, became 6 or above, which would suggest that, okay, that's not going to have a major influence on volatility because we are moving that pH upward, certainly much higher than 3 or 4 from our original pH measurements. Now we move to Xtendimax. Here you can see that, yes, certainly pH 3, we had up to 3 times greater volatility of dicamba compared to the other pH ranges. And there was no difference when we had a pH of 4 versus 5 or 6, all relatively the same. And yes, if we increased the pH even higher to 7 or 8, we had a further reduction in pH. But I want to remind you again that pH 7 or 8, we also have to be concerned about efficacy of the herbicide to control weeds. And there's some research being conducted in that area. But again, when we look at the leaf surface pH, even though we had a spray solution pH of 3, the deposit on the leaf surface within a minute after application was all 5 or above. And so this can explain why we didn't see as dramatic of an effect of having a lower pH on dicamba volatility at pH 4 or 5 because they're being buffered upward. Moving to Engenia, similar impact as what we saw with Xtendimax. That between pH 4 and 6, we didn't see any difference in dicamba volatility. It was similar to no pH adjustment at all, which was 6.4. We did see some reductions at the higher pHs, but again, caution we'd have to have if we might be attempting to control weeds with spray solutions that pH with dicamba, potentially. But we did see, again, about a 3x increase, 2.5x increase in volatility for Engenia when applied at a pH of 3. Looking at the leaf surface, again, even though we had lower pHs in solution that were adjusted that way in the lab, once on the leaf surface buffered, if you will, that pH so that it ultimately increased it to above 6. Why we thought it was negating some of these minor reductions in pH that we saw from the spray solution itself. So pH was having some effect, turbidity really not much of an effect. What about other spray solution ions that might find it into your spray tank? And these can be found, as your water source, hard water cations, ammonium sulfate, something that is not labeled for use with dicamba in extended crops, but in some cases might be found. We want to use that as a standard too, because it's bad and can increase volatility. Well, to what extent is that? Is that our benchmark for some of these other ions that might find it in the spray solution? So we want to look at some of these and test those. And again, the data format is fairly similar with the 3 herbicide formulations along the top and our treatments that we're making comparisons within in each column. I guess we'll start with the good news. The good news would be that regardless of the formulation, calcium sulfate, magnesium sulfate, calcium chloride had no impact on volatility. Now, the negative news is, yes, certainly ammonium sulfate did impact volatility by a factor of at least 12, and in some cases up to 16x increase in dicamba volatility when ammonium sulfate was used. So there's a good reason why we don't have that allowed on these dicamba labels for use in Xtend crops. One surprising aspect, though, is ferrous sulfate. Ferrous sulfate had a similar level of increase in volatility as ammonium sulfate. And we know it's not the sulfate component because we didn't see the increases with calcium sulfate or magnesium sulfate. So we know by itself, sulfate is not causing this increase in volatility. It is the iron and it is the ammonium. Ammonium makes some sense because we would think that that's going to provide a hydrogen ion. It's going to help acidify your spray solution. But the actual difference in solution pH by adding ammonium sulfate was only in the area of about 0.2 pH units. So you might take a spray solution of 6.3 and reduce it down to 6.1. Based off of our pH research, that's not significant enough to cause a major increase. Iron sulfate, that's where we have real interest in learning what more might be going on. So just to summarize the results in terms of the chemistry in the spray tank, turbidity of that spray solution had no effect on increasing volatility of dicamba. Spray solution certainly did, and we noticed that there's a buffering issue with the leaf surface. Now, if the pH was the soil pH, obviously the soil pH is going to buffer that spray solution as well once it hits that as a target. In this case, it's a leaf surface that's buffering it. In this case, it's buffering the pH a little bit higher than what our spray solutions were originally set to. And then in terms of the spray solution ions, both iron and ammonium sulfate increased volatility dramatically. Not a surprise for ammonium sulfate, but what is a little bit concerning here is we know that ammonium and ferrous are doing it because it's not the sulfate that is increasing the volatility. And I will mention that the iron concentration in the water supply that we sampled is much higher, or it should be the water supply that we tested was much higher than you would find in any groundwater, even in some of the hardest water you might find in your geography. But it is similar to at least one of the products we found that is approved for use in some of these Xtend formulations of dicamba that are approved as a foliar feed nutrient. So there is iron sulfate that are sold as a foliar feed with dicamba products that are currently on label to do so. And there's similar concentrations that we found this up to 16x increase in volatility. The one last thing I'm going to address in terms of volatility is environmental conditions. And again, Mandy did a fine job of this, talked about dew as a potential indicator of temperature inversions. But we know that dew was involved, has been implicated in being involved with increasing the presence or observations that dicamba volatility might occur. And so we want to know, is it the dew that is on the target leaves that you sprayed with dicamba that's important to have, or is it dew on the sensitive soybean plants that might be injured by dicamba volatility? Which component is important? And so we designed an experiment where we had these hoop houses and we simulated dew. We knew the pH of the dew. We simulated that. We knew the amount of dew in terms of how much water per hectare per acre needed to be sprayed on those at different time intervals. And we had simulated dew at 3 continuous nights. And I'm just going to go quickly to the results. You can see that when we had dew present, soybean injury increased. Okay, so soybean injury on the sensitive plants increased when dew is present on those, and then also on the sensitive plants, we also saw this increase. So when we had dew present on the target leaves that were sprayed, as well as dew present on the sensitive plants, in both cases, we saw an increase in the injury response that we measured in these low tunnel situations. So to summarize that, dew increased the potential for volatility as well as the potential for injury of the sensitive plants in our research. And let's bring this down to overall conclusions. Water turbidity doesn't seem to be a factor in influencing volatility. Certainly should be a concern though when we're trying to control weeds and herbicide efficacy, because some of that sediment can tie up herbicides. Leaf surface pH is buffering the spray solution or deposit of that spray solution on the leaves, so it didn't cause as big of an effect as what we would hypothesize. Now, one thing that I need to mention, we did not test these dicamba formulations in the presence of Roundup or other glyphosate products. We know that glyphosate can reduce the pH down to typically a pH in the low 5s, and it probably also provides more of a buffer so that it's going to resist any change in an increase in pH once on the leaf surface. So that's something we're going to investigate with some future research. And then also, as mentioned, there's been research out of University of Missouri that has shown that soil pH can increase volatility if you have lower pH soils, at least the surface soil pH, which is where that dicamba deposit is going to be applied. And then we also saw the increase in volatility from iron and ammonium sulfate. We know or think we know about ammonium sulfate. Iron sulfate, we're still wondering why that is increasing volatility. Must be something chemically, of course, but we're conducting more research to elucidate that in the future. And then lastly, dew is a contributing factor. It's a minor factor. It is minor, but it is another one of those factors where if we saw 10% injury on our soybeans, it might have increased it to possibly up to 20% injury on our soybeans. So it's not the major factor, but something that we also need to consider. So with that, Mark, that completes my presentation. I think I'll turn it over to you to help field some questions.
Great, thanks. Mandy and Bill, if you want to unmute, we can handle the questions and do a few things. Bill, I don't know if you got any emails via or any questions via email.
None yet.
Okay.
We did have a couple of questions that you and I came up with as they were speaking. So, Mandy, I've got a question for you about some of the applications that we can use on a smartphone for detecting inversions. Can you maybe give us your opinion of those and your opinion of, well, do they work, and is there some good utility with these smartphone apps?
Sure. So there are two types of, I'll just break down into two types of apps. One is current conditions. Is there an inversion forming right now? Or is there an inversion right now? And the other are the forecasting apps. And what I'll say about the real-time inversions is those apps are pretty reliable. They're measuring air temperatures right now. But as I showed that Tennessee site, a lot of it is field dependent. And so if you're looking at a app that is giving you whether an inversion is present 30 miles away, that might not be relevant to where you are. And so knowing the indicators of an inversion is just very important. For the forecasting apps, I've tested a couple of them out. And for some locations, basically I use our weather stations and determine whether those apps forecasted correctly at the weather station. And for some of those apps, or for some of the locations, those apps do really well. Again, for other locations, those apps don't do well. And it's not a fault. What they're trying to do is very hard. It's very hard to predict inversions at low levels near the ground. And I expect those apps to get better over time, but I recommend just knowing the signs of an inversion. And maybe smoke bombs can be a helpful visual.
Thanks. We do have a question that came in on the question box, which is, what is the influence of spray volume on volatility? That is 15 versus 20 gallons per acre. Probably for you, Bryan.
Yeah, good question. So in terms of spray volume, carrier volume, 15 versus 20, I don't believe there's any research that would suggest there are problems with increasing carrier volume. I actually did some research. I'm trying to recall that from a couple of years ago. But if you think about it, it's actually reducing some of the, it's going to have less of an effect on pH if you have higher carrier volumes. Your pH is going to be driven by your spray water that you originate from, and to a lesser extent by anything you're adding to that tank. So I don't think increasing the volume to 20 GPA is going to increase volatility. I think it probably worked the opposite way, that having lower carrier volumes will increase your chance for volatility.
Great, thanks. Another question that came in was from Mandy. Was the EPA graph of herbicide applications for all herbicides? Well, was it for all herbicides or specific to dicamba applications?
Yeah, that's a good question, and it was for all herbicides. You know, you have to look at it and say, okay, well, if a lot of herbicides are applied here, then perhaps there's more dicamba in this area versus other areas. What we know from Missouri, the information that we can obtain, that map does kind of parallel what we see in some geographies for dicamba as well. Thanks.
Yeah, that would make sense. I mean, if you have a whole slug of applications going on, that dicamba would certainly be part of that, I would think. Follow-up question, probably for you, can we come up with cutoff application timings for regions based on the research?
I'd be curious of Bryan's thoughts as well. I think maybe we're getting there, but I'm not sure we're there yet. Bryan, what do you think?
Well, thanks for passing that one off. So yeah, we're starting to learn more. I mean, if we had, you know, cutoff dates really addresses, well, how many complaints are acceptable and how many aren't. And so some state regulatory agencies are making that decision. Is the cutoff date going to be based on, you know, some environmental factor that they know about or is going to increase after that date and is going to promote more off-target movement? Well, I think temperature is the one that comes to mind. So, you know, to some extent, I want to say that maybe temperature cutoff is better than a calendar date. You know, temperature cutoff has been listed on some other dicamba labels. So I don't have a great answer for that. I think certainly it could help address some off-target movement issues if we limit how many can be applied later in the season. But from a scientific standpoint, you know, it just depends on each situation.
Yeah, I think it's an interesting comment about, so what's an acceptable level of complaints? You know, here in Ohio, we just, you know, the general levels not is low enough that, you know, I mean, the Department of Ag's not going to do anything. So I think, you know, that's an interesting take on that. I think one of the questions I had was, given all of this, can you make a general, I mean, I think about times in the summer when it's, you know, we're just hitting those warm nights and just hitting periods of that type of weather where you could, I know this is it's a hard question to answer, and it's not fair to you, but I mean, in terms of identifying, hey, we're coming into this type of weather pattern for the next week or the next 3 days that would really be conducive or more conducive to movement.
Our climatologist talks about this a lot, and you're right, Mark. At least for the inversions, you know, there are you'll have 4 or 5 nights in a row where it's going to be clear nights and no clouds, and so that's what he calls a persistent forecast. And you know, so kind of planning for that if it's possible, but it's so hard to plan for that. You know, that's asking a lot of applicators too.
Right. I still, yeah, I guess if we could identify that a little bit more closely, I think that would be helpful. I did have another question about the rainfall, because I think the current restriction on the label still is you cannot apply and it's a measurable rainfall or a certain chance of rainfall occurs within the next 24 hours. Is that still right, everyone? I think that's right.
I think it's saturating, yeah, a saturating rainfall.
Which would sort of, and I've seen it move. I mean, I don't remember exactly what the interval was, but I have seen it move with runoff water and pond in areas and cause damage to soybeans. So that works against your research a little bit in terms of rainfall being beneficial in terms of reducing the potential for movement.
Yeah, I think one of the things, you know, in research, you're always trying to think, okay, what can we do next time, or what can we do better? And I think one of the things would be, how much rain do we need to see that effect? We did a 1/2 inch, 6 hours after we sprayed dicamba in that research. You know, could you do less than that? I don't know. And in some areas that was saturating because it had been raining a lot, and in other areas that wasn't saturating to the soil.
Right. That which would certainly determine whether the potential, I mean, you have dry soil, you're going to have to take a lot of rain before you get it moving with runoff water versus soil that's already wet for more persistent rainfall, right?
Right.
Bill, do you see other questions? I don't, that's all I see.
I just had one other one here. Bryan, given the impact that pH has on volatility, spray solution pH, what should we be doing there? Should we be checking it? Should we be adjusting it even without checking it? What's your recommendation?
Well, I think we certainly should be checking it. I mean, there's no harm in doing so because we also need to check it to make sure we're optimizing herbicide efficacy too, where pH is important. You know, you've published on water quality and how it influences just weed control. So I don't think it's very difficult to check the spray pH. I think it's important. And, you know, fact is, what I'm going to guess here, I could be completely wrong, maybe 90% of all dicamba applications applied to extend crops contains glyphosate, you know, like Roundup Power Max. And that is going to alter the pH and probably buffer it more than what my pH adjustments did in the lab setting. So it's something else to consider, you know, where your spray pH is at. And then I think, you know, checking out this iron sulfate, that's not a pH adjustment, a major pH adjustment, but there still is increasing volatility. So simple answer is yes, I think we should check the pH.
Right, and we just had a question. I think you answered the question. I did something that I thought about was you did that work on soybeans, right, in terms of the pH on the leaf surface.
Yes.
So I mean, if you would you apply that to weeds, like if you sprayed you know, a dense stand of foxtail or something?
I would, and the reason I say I would is because you know, I have this big binder of that's the leaf surface of major weeds, and in that they analyze the pH of the leaf surface as well. That's where I got my methods for measuring soybean leaf surface pH, and most of those weeds it's fairly neutral, around 7. You know, maybe plus or minus half a pH unit, so 6.5 to 7.5. So I do think we'd have similar response on other target plants or off-target plants.
Okay, thanks. Great. Anything else? Bill, you have anything?
I don't have anything else on this end.
Great. I want to thank Mandy and Bryan for great presentations, and thanks to all of you who submitted questions. That wraps up today's presentation. You can get a recording of this webinar on the Take Action website within a week. And this also wraps up the series. Thanks to all of you who have tuned in over the past 7 weeks on behalf of myself, Bill, Take Action program, all the speakers, and everybody else who contributed. We appreciate your support. We intend to try to do this again next year at some point. We would certainly welcome your feedback and questions on any of the topics or any suggestions you have for how we might modify that. As a reminder, you can find recordings for all of the webinars at www.iwilltakeaction.com/management. We want to wish you a good season. A lot of us at the university are sort of in holding patterns right now. So we would certainly wish that you all get through this this current thing going on to the best, and we wish you well. Thanks.